Sure. So I'm John Simons, Professor of Cognitive Neuroscience here at the University of Cambridge, and it's my great pleasure to introduce Samantha Mitchell, who's in the second year of her PhD, supervised by myself and Dr. Jane Garrison. Sam was an undergraduate at Cardiff, where she studied for a BSc in psychology. She stayed on there for a master's as well, for which she received a distinction. And then we were lucky enough to recruit her to Cambridge, where she's worked very hard and made great progress so far on a number of different PhD projects focusing on the neurocognitive basis of hallucinations in schizophrenia, which we'll tell you all about now. So over to you, Sam.
Okay, thank you. I'm Sam. I'm a second year PhD student, and my PhD looks at the brain mechanisms of hallucinations with a focus on reality monitoring and the medial prefrontal cortex. So hallucinations are one of the many debilitating symptoms that can affect individuals with schizophrenia. And there are approximately 21 million people in the world living with schizophrenia. And it's estimated that around 70% of such individuals experience hallucinations. Hallucinations can occur in a range of modalities, so you have visual, tactile, olfactory, but the most common type by far are auditory verbal hallucinations. This is where individuals hear voices and sounds that are commonly distressing in nature, but in the absence of any speaker or auditory source. And despite great research interest into hallucinations, there is still no unified understanding of the causal factors underlying their manifestation. So I'd now like to introduce you to reality monitoring. So reality monitoring is the ability to discriminate between information that is internally generated, so your imaginations, your dreams, your thoughts, from information that is externally generated, so things that are actually happening in reality in the world. Have you ever thought to yourself, did that really happen, or did I imagine it? Well, we make those judgments every single day without even consciously knowing, and that is reality monitoring. Just think to yourself how important it is for you to be able to know that what you're experiencing is real and be able to tell the difference from your imagination. And why is that important here? Well, one of the many theories regarding the cognitive mechanisms underpinning hallucinations is that in schizophrenia, they may arise in part due to a deficit in this reality monitoring ability. So where sensory information, such as your inner speech or images, are perceived to be generated by an external source, and so they're perceived to be occurring in the outside world, and this would be the hallucination. And compared to healthy individuals, patients with schizophrenia have been found to perform worse on tasks of reality monitoring, and in patients, such deficits in reality monitoring have been associated with the presence of hallucinations. So the medial prefrontal cortex is an area of the brain that has been repeatedly associated with reality monitoring and hallucinations, and I've highlighted this area here in blue. And differences in this area relate to individual differences in reality monitoring across both clinical and non-clinical populations. But importantly, reduced activity in this area has been associated with poor reality monitoring performance in patients with schizophrenia. Within this area, research has highlighted a particular structure, the paracingulate sulcus. The paracingulate is a fold within the medial prefrontal cortex, and it's one of the last folds to develop in gestation. And it can vary quite considerably. So it can be short, long, deep, shallow. It can be split into many smaller sections, or sometimes it's not even present at all. And differences in this fold's morphology have been associated with reality monitoring performance. So bilateral absence of the sulcus, and this is where it's not present in either hemisphere, has been associated with poor reality monitoring performance in healthy individuals. And interestingly, it was shown that a 1 centimeter reduction in the length of the fold was associated with a 20% increase in the likelihood that patients with schizophrenia experienced hallucinations. So my PhD can be summarized into 3 main areas or themes. So first we have cognitive, so this would be abilities such as reality monitoring. And then we have structural MRI. So this is looking at the size, the shape, the different structural aspects of the brain. And then we have functional MRI. So this is the MRI that can tell us about activity or lack of activity in different areas of the brain. And I'll now take you through how I'm currently tackling each of these. So cognitive. The first aim of my PhD is to further investigate this link between reality monitoring and hallucinations or hallucination proneness. In particular, whether this link is modality general or modality specific. So what I mean by this is that there are many different types of hallucinations, with auditory hallucinations being by far more common than the rest. But are all of these underpinned by a global reality monitoring deficit, or are auditory hallucinations associated with auditory-specific reality monitoring deficits, and so on? In the literature, there wasn't a task sufficient to tackle this question, and therefore I spent a good portion of my first year designing and piloting a new task. So reality monitoring ability is commonly assessed using tasks that use word pairs, and here is an example. So participants are either presented with a complete word pair, so an example could be Laurel and Hardy, or they are presented with incomplete word pairs, so here fish and if it's incomplete, they're required to imagine that second word. So here they'd need to imagine the word chips. And later they are assessed on whether they can correctly recall whether they perceived or imagined that second word of the pair. So I adapted this commonly used reality monitoring paradigm into two distinct tasks that can measure visual and auditory reality monitoring abilities separately, but in a way that is directly comparable. So here you can see an example of each of the tasks. In the visual task in the study phase, you are presented with either complete or incomplete celebrity or character pairs. And when the pair is incomplete, the participant is required to vividly imagine the individual that would complete the pair. So here you either see ant and deck, or you just see ant, and you'd be required to imagine an image of deck. And then in the test phase, participants are asked whether that second individual was imagined or seen. And then in the auditory task, participants hear either complete or incomplete word pairs. So here, if they heard Romeo and, they would be required to imagine the spoken word Juliet. And later, they are required to make the same reality monitoring judgment. We've conducted this new task on around 75 healthy young adults so far, and I'm continuing to conduct large-scale online data collection. And then I will correlate these findings with modality-specific hallucination proneness scores. And this will allow us to see whether visual reality monitoring is associated primarily with visual hallucination proneness, and so on. Then secondly, we have structural. So I have introduced you to this parasingulate sulcus. And the second aim of my PhD is to analyze whether this association between psychosis, reality monitoring, and the length of this sulcus is present in younger ages, such as adolescence and childhood. And if it is, could we use it as a predictive marker for later psychosis development? Now, due to the great variability in this fold, it can be quite difficult to identify and measure. So a lot of the key research in this area was conducted measuring the sulcus on 2D MRI images, as you can see here. So here, the parasingulate sulcus was manually traced with this red line onto this 2D image. But now technology is allowing us to use 3D models of brains, and these here were produced in a software called Brain Visa. And it's provided us with 3D representations of the sulci. So I've identified here the parasingulate sulcus and colored it in green. And you can see that it's present in the first individual's brain, but absent from the second. And this change from 2D to 3D is already a great improvement and provides us with so much more information. So such as the depth of sulci, but also you can interact with the images, twist it around, rotate, and really have a look at what's going on in the brain. But even in these 3D models, identifying such a highly variable structure is still very challenging. So therefore, a colleague and I have spent some time this year developing a list of updated guidelines that will allow researchers to more accurately and reliably identify and measure this parasingulate sulcus using this 3D software. So it became clear that the best way to identify this parasingulate sulcus was actually to first identify the other less variable sulci in the area. So along with in-depth guidelines for the expected location and trajectory of each of these other sulci, the protocol provides a specified order for sulci identification. So first, you would identify the callosal sulcus. This is the sulcus that lies right over the corpus callosum. And then you'd identify the cingulate sulcus. This is here in blue, and it's a primary fold, which makes it much less variable in its structure than the parasingulate sulcus, making it much easier to identify. And then if there are any sulci above and parallel to the cingulate sulcus that match all other specifications of the parasingulate, we would then identify these as elements of the parasingulate sulcus. And a recent validation of this protocol showed that we have very high agreement between raters when using it, and we really hope that it will be useful for researchers in this area. The next step will be to use this new protocol to look at paracingulate sulcus in adolescent datasets. So first, I'm looking at the NSPN cohort. This is the Neuroscience and Psychiatry Network. It's a cohort of around 2,400 14 to 24-year-olds, and a subset was scanned a few years ago. I have been granted permission to recontact the individuals to complete a reality monitoring task, and then I will see whether the paracingulate sulcus length from MRI scans conducted during their adolescence can predict their current reality monitoring ability and their psychosis status. And then following on from this, I will also look at the ABCD cohort. This is the Adolescent Brain Cognitive Development Study, and this is a much larger database of around 12,000 individuals who entered the study at around 9 to 10 years old. And here I will look at the paracingulate sulcus length of children and compare between children who are at the most and the least risk of psychosis development. And thirdly, we have functional. So as I've said, activity in the medial prefrontal cortex is associated with this reality monitoring performance and hallucinations. And technological advances has provided this technique called fMRI real-time neurofeedback. And this technique has been found to allow individuals to selectively increase activity in certain regions of their brain following some training. And this has been shown to lead to changes in the cognition, the behavior, or the symptoms observed by that targeted area. And a recent neurofeedback study conducted from our lab found that active neurofeedback training targeting this medial prefrontal cortex in healthy individuals led to increased activity in the area. So I'd like to build on this study using updated techniques and software and see whether neurofeedback increases reality monitoring performance, firstly in healthy individuals, but then in patients who experience hallucinations. And if it is feasible to run such a session in patients, and if reality monitoring ability is improved, future steps could look at how long-term this effect is and whether this non-invasive technique could reduce the severity or the frequency of hallucinations. And thank you very much for listening, and thank you to Mental Health Research UK.